Hi, I'm Jamin, and this project aims to design drugs for COVID-19 using generative RNN-LSTM model. The aim of this project is to design potential drug candidates that can bind to the COVID-19 main protease. So basically, the COVID-19 main protease, also known as SARS-CoV-2 main protease, acts like a lock, and we are trying to design inhibitors that can bind to this lock and hopefully stop the whole virus life cycle process. Now, to be able to do this, we can first get dataset and train them using RNN-LSTM model to generate new molecules and fine-tune them using transfer learning. Then we can use molecular docking technique to get the binding affinity between the molecules and the COVID-19 main protease. Now, first of all, we can get dataset from Campbell and Singh, which has all the molecules with drug-like properties. The molecules are represented as a SMILE format, which stands for Simplified Molecular Input Line Entry System, which is basically a string like this shown here. This dataset was then pre-processed to remove duplicates, salts, stereochemical information, long peptides, and all the things that we don't want. This string can then be fed into the model for the model to train and learn and to generate new molecules, new strings. PCA was performed to compare the generated molecules and the original molecules. So the generated molecules are shown here as the red color, and the original molecules are all the circles. Both of them are in the same chemical space, which is a good thing. The performance of the model was evaluated, and the validity, uniqueness, and originality are all pretty high, which means that the molecules are not duplicated and they are valid SMILE strings, they are valid molecules. The next step is to fine-tune the model using transfer learning. What this means basically is that we put in the known protease inhibitors for the model to learn and fine-tune so that it can generate molecules that are structurally more similar to these known molecules. And the generated structures are shown on the right. All these are the closest neighbors to the known molecules, which was calculated using Tanimoto similarity included in the RD-Kit package. So the last step is to get the binding scores between these molecules and the COVID-19 main protease. This is a snapshot of PyRX software that I used to get the binding scores from. I've included a reference link on how to use this in my README and Python notebook, so if you're interested, you can go and check it out. So here's the final result of the binding affinity between the molecules and the COVID-19 main protease. Lopinavir shows the highest binding score with negative 6.9 kcal per mole, followed closely by Generative 3 with negative 6.8 kcal per mole. The more negative the binding affinity is, the better it binds to the protein. So the more negative, the better it is. In conclusion, the generative RNN-LSTM model was able to generate new molecules that can bind to the COVID-19 main protease. So I've uploaded all of my work to GitHub, so if you are interested to know more, feel free to click on the link and check it out. So here's the Jupyter notebook that has most of the work that I've done, and it looks like this. I've tried to make it as clear as possible, and it's like a report format. So I hope it's clear, and yeah, that's how we can use deep learning to design new drugs for a disease in a more efficient way. Thank you.